Hey everyone, and welcome to this explainer. Today, we're going to challenge a pretty common misconception. When most of us hear the word scientific method, we instantly picture, white lab coats, super complex chemical formulas, and these rigid rules confined to sterile laboratories. But we're going to completely reframe that idea today. The scientific method isn't just some intimidating academic requirement. It's actually a powerful, everyday mental model for problem solving. Honestly, it's a toolkit you are very likely already using without even realizing it. And once you see the underlying structure of how we optimally solve problems, it genuinely shifts how you approach the world.
Okay, let's dive into this. Here is our roadmap for the explainer, and it logically follows the exact sequence of a classic scientific investigation. We'll start with a scientific problem-solving toolkit, then identify the three variables, formulate a hypothesis, test and gather data, and finally, draw meaningful conclusions. Let's jump right into step 1. Section 1, the Scientific Problem-Solving Toolkit. This is all about applying science to everyday life. Now, what's really interesting about this is that every great scientific investigation throughout history starts the exact same way, with a simple observation of a problem or some frustrating bottleneck. It doesn't have to be a multi-million-dollar physics equation. It can be a classic optimization problem. Imagine teacher Mara trying to prep a classroom wall for a fresh coat of paint, only to hit a really stubborn bubblegum stain. This isn't just a chore, it is our very first test case. The scientific method is triggered the literal moment you look at a problem like this, ask yourself, how do I fix this efficiently, and seek a systematic way to find the best possible solution. Moving on to Section 2, identifying the three variables. Let's break down the moving parts of an experiment. To solve our sticky bottleneck, we can't just throw things at the wall randomly, right? We need to understand the specific elements we can control, the elements we deliberately change, and the elements we want to observe. In the scientific community, we call these moving parts variables. And distinguishing between them, that's the absolute secret to running a fair and accurate test. First up, we have the independent variable. Think of this as the cause. It's the one condition you are actively manipulating. Next is the dependent variable, or the effect. This is the condition that changes in response to your independent variable. It's what you're actually measuring. Finally, we have the controlled variables. These are the conditions kept exactly the same throughout the test so they don't skew your results. Without controlling your environment, you really won't know if your independent variable actually did the work. So, to solve teacher Mara's bubblegum bottleneck, we have to ask a question. Which material removes it best? Are we using oil, water, or ice? By deliberately choosing different materials to test, the material itself becomes our independent variable. The dependent variable, the actual effect we're measuring, is simply how easily that gum comes off the wall. And this brilliantly illustrates a logic we see absolutely everywhere. Take pandemic-era indoor gardening, for example. Consider a plantita mom planting tomatoes. Some go outside, into garden soil with full sunlight, while others go into small pots inside the house. The independent variable, the isolated change, is the location of the plants. But for a fair scientific test, controlled variables must be utilized. You have to give both sets of plants the exact same amount of water and the exact same type of fertilizer. By controlling the water and nutrients, any difference in growth, which is your dependent variable, is strictly due to their location. Which brings us to Section 3, formulating your hypothesis. It's time to make an educated guess. Once you've identified your problem, and clearly mapped out those variables, you don't just jump straight into testing. You pause, you tap into your existing knowledge, and you predict the outcome by formulating a hypothesis. And writing a solid hypothesis is incredibly straightforward if you use 3 concrete steps. Step 1, define the problem clearly. Step 2, determine your variables. Step 3, phrase your guess as an if-then statement. This specific rhythm is vital, guys. It forces your brain to directly connect your independent variable to your dependent variable, creating a strictly testable sentence. For example, if ice is used, then the bubblegum stain will be removed. Notice how elegant and perfectly testable that is? It perfectly links the independent variable, using ice, with the expected dependent variable, the removal of the stain. It totally avoids vague assumptions like, I think ice is kind of good for cleaning. Instead, it provides a strict measurable condition. If X happens, then Y will follow. Next up, Section 4, testing and gathering data. We are putting the hypothesis to the test. Now, let's move to and see how this builds into the real world. We have our if-then statement, and now we actually have to run the experiment to see if reality behaves the way we predicted. Take Manuel, a farmer dealing with mice destroying his crops, lowering his harvest, and really affecting the local food supply. Manuel hypothesizes that peppermint might act as a deterrent. So the experiment is designed with the independent variable being the planting of peppermint right in between the rice crops. From there, it is just a matter of waiting, testing, and gathering data on the mouse population. But here's a question for you. How do we objectively know the experiment worked? Well, data gathering is simply the act of objective observation. You are measuring whether the dependent variable reacted the exact way your hypothesis predicted. For Manuel, a farmer, data gathering involves literally counting the mice or measuring his crop yield. If the crop yield goes up and the mice population goes down after introducing that peppermint, boom, the data supports the hypothesis. If the mice just keep eating the rice anyway, well, the data objectively tells him the experiment didn't work the way he hoped.
And finally, Section 5, drawing meaningful conclusions. Let's make sense of all that data. You've got your numbers and your observations, but numbers alone aren't an answer. You have to look at the results and make a definitive decision. Was your initial educated guess correct, incorrect, or inconclusive? As Stephen Novella brilliantly points out, science is about the process. It's not about the conclusion. This is deeply important to remember. Sometimes you test a hypothesis and the data proves you completely wrong. The ice might not remove the gum. The peppermint might not deter those mice. That is absolutely not a failure. A disproven hypothesis is incredibly valuable scientific data. The method worked perfectly because it successfully taught you what doesn't work, which guides you right closer to the truth. So, the crucial point is that this framework scales effortlessly from household chores to massive global events. During the pandemic, for instance, officials needed to gauge the impact of quarantine levels on infection rates. The problem? Are there more infections under General Community Quarantine, or GCQ, versus Modified Enhanced Community Quarantine, or MECQ? The hypothesis? More people are infected under the looser GCQ. The independent variable? The quarantine level itself. Data gathered? Purely objective numbers, like 200 cases under GCQ versus 150 cases under MECQ. And the conclusion? The data supports the hypothesis that more people were infected under GCQ. Whether you're dealing with a sticky wall, hungry mice, or critical public health data, the framework for extracting truth from observation remains exactly the same.
To really solidify this mental model for yourself, I want you to ask yourself a few questions. Take a moment later today to mentally jot down 3 things you learned about the scientific method, 2 things you now wonder about the variables floating around in your own daily routines, and 1 question you still have about drawing unbiased conclusions from data. Processing the information this way is exactly how you firmly lock it in.
I'll leave you with this final provocative thought. What everyday problem will you solve with the scientific method today? Because you now have the exact same problem-solving toolkit used by the greatest minds in history. Go observe a problem, identify your variables, write out that if-then hypothesis, and put it to the test. Thank you so much for joining me in this explainer, and happy experimenting.